Assalamu alaikum and welcome everyone to the second part of our molecular biology ASCP preparation series, Molecular Techniques. I'm Dr. Lay Abdalati and I'm really excited to share my knowledge and experiences with all of you through this series. So let's dive in together. Today, we'll explore one of the most essential tools in molecular biology, DNA sequencing, specifically Sanger sequencing and next generation sequencing. This method allows scientists to read the genetic code, helping us understand genetic variation, diagnose disease, and advance personalized medicine. So, welcome to the session.
Break it down small like DNA. Learning the code where secrets stay. Chromosomes dance under the light. Molecular dreams will take their flight. Molecular magic, it's in your hands. Unlock the science and take a stand. Study hard, let your passions ignite. ASCP success is within your sight.
Now, let's move to the question section, and I will start with the Sanger sequencing. First, Sanger sequencing, developed in 1970, is a trusted method for sequencing smaller DNA fragments with the high accuracy. It works by using a special nucleotide that terminate DNA replication, creating fragments that reveal the sequence when analyzed. It was used in research and clinical setting when precision is key. All right. Let's see the first question we have today in Sanger sequencing. What is the key enzyme used in Sanger sequencing? The correct answer is DNA polymerase. In Sanger sequencing, what causes chain termination during DNA synthesis? The correct answer is DDNTBs. Actually, DDNTBs lack of 3' hydroxide group, preventing the addition of more nucleotides, thus terminating the DNA strand during synthesis. All right? Okay, next question here. In Sanger sequencing, which type of capillary device separates DNA fragments by size? The correct answer is capillary electrophoresis. Actually, capillary electrophoresis separates DNA fragments based on size by running them through a thin capillary tube under electrical field, allowing precise detection. All right, let's move on. Next question. In Sanger sequencing chromatogram, what does a sharp single peak represent? The correct answer is a single correct nucleotide base. All right. Actually, each sharp peak corresponds to successful reading of a specific nucleotide, ensuring accurate base calling. All right. Let's move on. Okay, the next question here. Why are DDNTBs used at low concentrations in Sanger sequencing reactions? All right. By using low amount of DDNTBs, random chain termination occurs, producing fragments of different lengths need for sequence termination. All right. So the correct answer here is to allow chain termination at random positions. Let's move on. Okay, the question here. Which discovery made Sanger sequencing widely automated? The correct answer is fluorescent labeled ddNTBs. All right. So the use of fluorescent dyes attached to the ddNTBs enable automated DNA sequencers to read results quickly and accurately. All right. Let's move on. All right. What would be happen if no ddNTBs are added to the Sanger sequencing reaction? For sure, without ddNTBs, DNA polymerase would continue copying the template without termination, leading to continuous full-length DNA strands and preventing sequencing result. All right, let's move on. The question here, why is only one primer used in traditional Sanger sequencing? The correct answer is to generate single-strand DNA fragments. All right? All right. What is the major limitation of traditional Sanger sequencing? As we know, Sanger sequencing provides highly accurate results but has low throughput, as it can only process one DNA fragment at a time, making it inefficient for large-scale projects. All right, let's move on. All right, how are DNA fragments detected in modern automated Sanger sequencing? The correct answer by fluorescent detection. All right. Actually, modern Sanger sequencing relies on the fluorescent dyes attached to the ddNTBs to detect and read the DNA sequence automatically. All right, let's move on. Okay, which of the following describes the output of Sanger sequencing reaction? The correct answer is a series of DNA fragments ending with the labeled nucleotides. All right, let's move on. All right, here is the last question in the Sanger sequencing part. What is typically the length range of readable sequence in Sanger sequencing read? The correct answer is 500 to 1000 bases. All right. Please, if you found this content helpful, don't forget to like, share, and subscribe. Your support means the world to me and help me create more valuable content for you. Thank you for being part of this journey. Now, let's move to the next generation sequencing part. And let's see the first question we have here. The question is, what is the main advantage of next generation sequencing over Sanger sequencing? The correct answer is massively parallel sequencing. It means next generation sequencing can sequence millions of DNA fragments simultaneously, making it much faster and more efficient than traditional Sanger sequencing. All right? What is typically the first step in next generation sequence workflow? The correct answer is DNA fragmentation. Actually, DNA fragmentation is usually the first step to break the DNA into manageable pieces that can then be prepared into sequencing library. Okay, let's move on. And Illumina sequencing. What does sequencing by synthesis mean? Okay, in sequences by synthesis mean each nucleotide added during DNA synthesis emit a detectable signal, allowing the machine to read the sequence as it being built. All right. So the correct answer here: each base addition is detected during DNA synthesis. All right. And next generation sequencing, what is typically attached to the ends of DNA fragments to prepare them for sequencing? The correct answer is adapters. Actually, adapters are short DNA sequences ligated to both ends of DNA fragments, allowing them to bind to the flow cell and be amplified during sequencing. Okay? Okay. What type of data is primarily generated by using next generation sequencing platforms? Okay, most of next generation sequencing platforms produce short DNA reads, often around 100 to 300 base pairs, all right, which are later assembled into longer sequences. Okay, let's move on. Okay, the next question here. Which sequencing platform is known for using bridge amplification? The correct answer is Illumina, all right? Actual Illumina technology uses bridge amplification to generate clusters of identical DNA fragments directly on the flow cell. Let's move on. In ion torrent technology, what is the detected to determine the added nucleotide? The correct answer is pH change. All right. Actually, in ion torrent sequencing, the incorporation of a nucleotide releases a hydrogen ion, causing a pH change that the machine detects. The next question here: which of the following best describes the flow cell in next generation sequencing? All right. A flow cell is a specially treated glass surface with the channels that allow DNA fragments to bind, amplify, and be sequenced. All right. Let's move on. Okay, next question here. What is the purpose of clonal amplification in next generation sequencing? All right. The purpose of clonal amplification in next generation sequencing is to create multiple identical copies of each DNA fragment, allowing for strong detectable sequencing signals in next generation sequencing. All right. Okay, the next question is, what is the multiplexing in the context of next generation sequencing? Multiplexing in next generation sequencing enables simultaneously sequencing of multiple samples in a single run by assigning each samples a unique barcode to distinguish them later. All right, let's move on. All right, the question here, which next generation sequencing platform is known for extremely long reads but higher error rates? The correct answer is X4 nanopore, okay? Actually, X4 nanopore sequencing can generate very long reads, even entire chromosome, but typically has higher error rates compared to the other platforms. All right, let's move on. Okay, next question. What is the one advantage of paired end sequencing over single end sequencing and next generation sequencing? The correct answer is better alignment and assembly. Actually, in paired end sequencing, both ends of each DNA fragment are read, which improve mapping accuracy and helps resolve repetitive regions. All right, the next question here. Which next generation sequencing platform uses real-time detection of DNA sensitivities without stopping the reaction? The correct answer is Pacific Biosciences. Pacific Biosciences uses real-time sequencing to detect nucleotide incorporation as the DNA polymerase sensitive the strand. All right? All right, the next question here. What happens if adapter ligation failed during next generation sequencing library preparation? The correct answer is the DNA fragment will not bind to the flow cell. As we know, without properly attached adapters, DNA fragments cannot anchor to the flow cells and will not be sequenced. All right, in next generation sequencing, what is the meant by the term coverage? In next generation sequencing, coverage refers to the number of times a base is sequenced. All right, let's move on. All right, the next question here. Which of the following is the common cause of sequencing error in next generation sequencing? The correct answer is homopolymer repeats. Okay, in next generation sequencing, homopolymer repeats like long stretches of same nucleotide like AAAA are common cause of sequencing error because they make it difficult for sequencing platform to accurately determine the number of repeated bases. All right, let's move on. Okay, which of the following best describe a paired end read in next generation sequencing? Okay, in next generation sequencing, paired end sequencing means reading both ends of DNA fragments. This provides more information about the fragments and improve alignment accuracy. All right. In Illumina next generation sequencing, how are different nucleotides detected? The correct answer is different fluorescent dyes. So each nucleotide, such as A, T, C, G, is labeled with a unique fluorescent dye, allowing the machine to detect which base was added during sequencing. Okay, let's move on. Okay, there is a question here. Which part of next generation sequencing process involves aligning the short reads to a reference genome? The correct answer is bioinformatic analysis. As we know, bioinformatic analysis comes after sequencing and include aligning the generated reads to a reference genome for interpretation. All right, let's move on. All right, the question here. Which of the following is the example of a next generation sequencing error correction method? The correct answer is using consensus sequences. All right. Actually, consensus sequences are generated by comparing multiple reads of the same region to reduce sequencing error and increase accuracy. All right. In Illumina sequencing, what is the index hopping? All right. Index hopping in Illumina sequencing occurs when barcodes get misassignment between samples, leading to incorrect read allocation and potential errors in multiplexed next generation sequencing runs. All right, let's move on. All right, the next question here. Which of the following would most likely cause low cluster density in Illumina sequencing? The correct answer is too little DNA loaded onto the flow cells. Actually, if too little DNA is loaded, few clusters form on the flow cell, leading to low cluster density and poor sequencing output. All right, let's move on. All right, this table provides a clear overview of major next generation sequencing platforms and their core sequencing principle. So, understanding this difference is the crucial for molecular biology ASCP exam. Okay, so each description is concise and highlight what make each technology unique. That's all for today. I hope you enjoyed this lecture. I look forward to hearing from you what you think of it. Please don't forget to like, comment if you have any questions, share and subscribe to our channel to receive all the latest lecture. And don't forget to click here to watch the previous videos. Salam.
Break it down small like DNA. Learning the code where secrets stay. Chromosomes dance under the light. Molecular dreams will take their flight. Molecular magic, it's in your hands. Unlock the science and take a stand. Study hard, let your passions ignite. ASCP success is within your sight.